Deep Dive with Mohio.
Welcome back to Deep Dive with Mohio. You know we love diving deep into all sorts of fascinating topics, but today we're focusing on something you deal with every single day as medical laboratory scientist, waste management.
Absolutely.
It may not be the most glamorous side of the job,
Right.
but trust me, it's absolutely crucial
Oh, it is.
for safety and for keeping things running smoothly in the lab.
And you know what's interesting? Most people would assume that lab waste is automatically hazardous. But that's not always the case.
That's surprising. What makes the difference then?
It all boils down to decontamination.
Yeah.
That's really the heart of safe and effective waste management. So today, we're going to break down how to sort and handle all those different types of waste that you encounter every single day.
Okay, so let's imagine I'm standing in the lab and I'm about to toss something out.
Yeah.
What's the first thing I should be thinking about?
Before anything leaves your lab, you need to stop and ask yourself 3 critical questions. Think of it as your waste management mantra.
Ooh, a mantra.
Yeah.
I like that.
Right.
It makes it sound almost philosophical. All right, hit me with these questions.
First one, has this item been properly decontaminated? Seems obvious,
Right.
but it's easy to get complacent, in a busy lab environment.
You're right. We can't just assume everything is sterile.
Exactly.
What's next?
If it hasn't been decontaminated, then you ask yourself, is it packaged correctly for incineration?
Okay.
Following all the regulations, we're talking specific procedures for the stuff that can't just be clean and needs to be completely destroyed.
So we're making sure that we're not putting anyone at risk during transport and disposal.
Absolutely.
Okay, what's that last all-important question?
Could this pose any risk, biological or otherwise, to anyone who handles it outside the lab?
That's a really good point. It's not just about protecting ourselves in the lab.
Right.
It's also about thinking of everyone else who might come into contact with that waste down the line.
Yes.
Big responsibility.
It is. And that leads us to the next crucial step.
Yeah.
Sorting. Think of it like a recycling system.
Okay.
But with a focus on safety rather than just, environmental friendliness.
Okay, so what are our main categories in this lab recycling system?
Let's break it down. First, you've got your non-contaminated waste. This is the easiest one.
Yeah.
Clean paper packaging, basic regular trash.
All right, so the stuff we can treat like regular garbage.
Yep. White containers for that.
Okay, perfect. What about the more serious stuff?
Then you've got your sharps.
Okay.
Needles, blades, broken glass, anything that could cause a puncture wound. It's all in the name, really.
Right.
These always go into those puncture-proof containers with lids and never overfill them. Yellow containers for those.
Right. So sharp's definitely a no-brainer in terms of needing that special handling. What about materials that have actually come into contact with potentially infectious material?
Right. So when we talk about contaminated waste, we have a few subcategories.
Okay.
First, those items that can be autoclaved.
To clarify for our listeners, autoclaving uses high-pressure steam to sterilize.
Yeah.
Like a super intense pressure cooker.
You got it. Exactly. So this type of waste goes into special autoclavable bags. You'll often see them color-coded for extra safety.
Yeah.
Makes things easier in a busy lab. And then after autoclaving, some items can actually be reused, which is a nice bonus.
Right.
But whether it's being disposed of or reused, it still ends up in those yellow containers.
Interesting. So both sharps and autoclavable materials go into the same color container.
Uh-huh.
I'm assuming the puncture-proof versus a regular container helps distinguish them.
Precisely. And there's another type of contaminated waste that goes in those yellow containers as well. Material that can't be autoclaved and needs to go straight to incineration.
Okay.
This is where those regulations we talked about earlier really come into play.
So it sounds like there's a lot of nuance to this.
There is.
So we've got non-contaminated sharps, autoclavable, and incinerate, only all going into yellow containers.
Yeah.
But with very specific bagging and container types to help further categorize them. Is there anything else in the contaminated category?
You've got a great handle on it. There's one more important category. Contaminated materials that are reusable.
Okay, that makes sense.
Yeah.
We don't want to be wasteful if something can be safely reused. We need to prep those before they go back into rotation.
You actually don't want to clean these reusable items before autoclaving.
Really?
I know it seems counterintuitive,
It does.
but trust me, cleaning beforehand could actually spread infectious material.
Wow. I never thought of it that way.
Yeah.
That's a good reminder that those safety protocols are there for a reason. So we've covered solids pretty thoroughly, but what about liquid waste? How do we handle that in the lab?
Great question. All those floor drains you see in the lab, they're not connected to the regular sewer system.
Oh.
They lead to a dedicated decontamination system,
Okay.
ensuring that potentially infectious liquids are treated appropriately before being released.
So there's a whole separate system for liquid waste that makes sense. Are there any specific procedures for particularly nasty stuff? Like, say, prion-contaminated waste?
Absolutely. For prions, you'd follow a specific protocol using a high concentration of sodium hypochlorite. The details are really important when it comes to ensuring those prions are completely inactivated.
Sodium hypochlorite, isn't that basically bleach?
It is, but we're talking about a much higher concentration than what you'd find under your kitchen sink.
Right.
It's all about guaranteeing those prions are completely neutralized.
Wow, that's some serious stuff. It seems like there's a lot to remember when it comes to waste management in the lab. What are some general best practices that our listeners should keep in mind?
One key point to remember is that disinfectant contact time is crucial.
Okay.
You want to make sure those materials are fully submerged in the disinfectant and that there are no air bubbles preventing contact. And of course, if you're reusing containers, always decontaminate and wash them thoroughly.
Air bubbles.
Yeah.
That's a great point. It's easy to overlook those little details. So to sum it up, it's all about being meticulous and following procedures to a T.
Yes.
No cutting corners when it comes to safety.
You got it. Every step in that waste management process, from sorting to disposal, is designed to protect everyone.
Right.
And ensure the integrity of the work being done in the lab.
This has been really eye-opening. I think we often take for granted how much goes into something as seemingly simple as throwing things away in the lab.
It's easy to get caught up in the day-to-day, but proper waste management is fundamental to everything we do in the lab.
Absolutely. And there's always more to learn.
Always.
We've covered a lot of ground in this first part, but there's still so much more to explore. Be sure to join us for part 2, where we'll delve into the specific regulations and protocols governing lab waste disposal.
We'll be back soon, so stay tuned.
Deep Dive with Mohu.
Welcome back to Deep Dive with Mohu.
Last time we covered all those different waste categories and the golden rules for handling them.
Right.
But now I want to talk about the regulations surrounding all this. I have to admit, the regulatory landscape can feel a bit overwhelming sometimes.
I hear you. Those regulations can seem daunting.
Yeah.
They're there for a reason. You know, they're all about protecting public health and the environment, ensuring we're doing things safely and responsibly. And remember, your lab will have specific guidelines to follow. Those should always be your go-to reference.
Okay, that makes sense.
Yeah.
So where do these regulations come from? Who's calling the shots here?
Well, there are national and international bodies that set standards for handling and disposing of biohazardous waste. In the United States, for instance.
Yeah.
You've got OSHA, the Occupational Safety and Health Administration.
Right.
And the EPA, the Environmental Protection Agency, playing key roles.
OSHA and the EPA. Those are some big names.
Yeah.
It definitely underscores the importance of taking this seriously. What kind of things do these regulations actually cover?
They cover a pretty wide range. Everything from how waste is labeled and packaged to training requirements for personnel and meticulous documentation of procedures. It's all about creating a comprehensive system to minimize risk at every step.
There's that documentation again. Seems like there's always paperwork involved in these important processes.
You're not wrong there, but that meticulous record keeping is really crucial.
Yeah.
It provides accountability and traceability. If any issues come up, maybe during an audit, you need to be able to demonstrate that you're handling waste responsibly and in accordance with the law.
Okay, so if I'm a medical laboratory scientist, what are some key things I need to be aware of in terms of my own responsibilities?
Well, first and foremost, you absolutely need to know your lab's specific waste management plan inside and out.
Okay.
That means understanding those different waste categories we talked about, the right containers to use for each, and the proper procedures for handling, labeling, and transporting waste.
Okay, so really knowing the system and how it all works in my specific lab. What else is crucial?
Personal protective equipment, PPE, is non-negotiable. Whenever you're handling anything that could be infectious, we're talking gloves, lab coats, eye protection, whatever's appropriate for the task at hand.
PPE, always important. It's easy to get complacent when you're busy.
Yeah.
But those precautions are there for a reason. Anything else in terms of individual responsibility?
You should be trained in the proper use and disposal of sharps,
Okay.
as well as what to do in case of a spill.
Right.
Accidents happen.
Yeah.
Being prepared is key to containing any potential hazards and keeping everyone safe.
Spills are definitely not something you want to mess around with. It sounds like training plays a big role in all of this.
Absolutely. Ongoing training is critical.
Yeah.
To ensure everyone in the lab is up to date on the latest regulations and best practices. It also reinforces how important it is to follow procedures consistently and meticulously, even when things get busy.
Consistency is definitely key. It's easy to get a little sloppy when you're doing the same thing day in and day out. So speaking of those day-to-day operations, let's talk about some specific procedures. We touched on autoclaving earlier. Can you break that down a bit more?
Sure. Autoclaving is that essential process for decontaminating reusable items and certain types of waste.
Okay.
It uses high-pressure steam to essentially nuke any microorganisms, rendering the material sterile.
So it's like a giant pressure cooker for lab equipment,
Yeah.
but with much higher stakes.
That's a great analogy. And just like your pressure cooker at home, autoclaves have precise controls for temperature, pressure, and time. Those parameters are critical to ensure complete sterilization.
And I imagine those precise parameters are super important for ensuring everything is actually sterilized.
They are.
Are there any specific guidelines for how to load and operate an autoclave correctly?
Absolutely. Overloading is a common mistake.
Yeah.
If you cram too much in there, steam can't reach all the items and sterilization could be compromised. You also need to use the right containers and indicators to confirm that the process worked as intended.
Indicators, what are those exactly?
They're tools that change color when exposed to those high temperatures and pressures inside the autoclave. Think of them like a little signal letting you know the sterilization process was successful. They can be tapes or strips, often placed inside the autoclave.
Okay, that's really smart. Takes the guesswork out of it. So once something has been autoclaved, is it safe to handle without PPE?
That really depends on your lab's specific protocols. Some items might need further handling, like washing or sorting before they can be disposed of or reused. Others might be ready to go straight into the disposal stream. Always check your lab's guidelines to be sure.
Right, no cutting corners when it comes to safety, even after autoclaving. What about incineration? You mentioned that some waste goes straight to the incinerator. What exactly does that process involve?
Incineration is basically a controlled burn at extremely high temperatures, reducing the waste to ash. It's a common method for getting rid of biohazardous waste, especially sharps and other materials that can't be autoclaved.
Sounds pretty intense.
Yeah.
What are some of the considerations for transporting waste to the incinerator?
It's not as simple as just tossing it in a bin and sending it up. The waste needs to be carefully packaged and labeled,
Okay.
often in specific containers or bags, to prevent any leaks or spills during transport. And there are regulations about who can transport biohazardous waste and how it needs to be secured.
So there's a whole chain of custody for this waste from the lab to the final disposal.
Exactly. It's about ensuring that the waste is handled safely and responsibly at every single stage. And it's not just about protecting people, but the environment as well. Incinerators have to meet strict emission standards to minimize air pollution.
That makes sense. We've covered a lot of ground here on the regulatory front. Any final thoughts on regulations and protocols before we move on?
I think it's key to remember that these are just general principles.
Okay.
The specifics will vary depending on where your lab is located, the type of lab it is, and the materials you're working with. Always consult your lab's safety guidelines. And stay in the loop about any updates or changes in those regulations.
That's great advice. We've covered all those different categories, the golden rules for safe handling, and even delved into the regulatory side of things.
Yeah, we've really gotten into the nitty-gritty, haven't we?
We have. But now I think it's time to zoom out a bit and consider the bigger picture.
I like that zooming out.
So how does all of this meticulous waste management connect to those larger principles of infection control and environmental responsibility?
Well, it's all connected. At the most fundamental level, proper waste management is all about breaking the chain of infection. By carefully containing, treating, and disposing of potentially infectious materials, we're not just protecting ourselves and our colleagues in the lab, but the entire community.
It's kind of like a ripple effect, isn't it?
Exactly.
Starting in the lab and spreading outwards.
Just imagine the consequences if a contaminated needle were to accidentally end up in the regular trash.
Oh, gosh.
It could injure a waste handler or even make its way to a landfill, posing a risk to the environment and potentially even impacting public health.
That's a scary thought.
Yeah.
It really drives home the point that every single piece of waste needs to be handled with the utmost care.
Absolutely. And it's not just about preventing the spread of infectious diseases.
Right.
Proper waste management also plays a huge role in protecting the environment.
We talked a bit about incinerator emissions earlier. What are some other environmental considerations that come into play?
Well, for one, we need to be conscious of the sheer volume of waste we generate. That means reusing and recycling whenever possible.
Okay.
Think back to those reusable containers and glassware we discussed.
Right. It's just like that reduce, reuse, recycle mantra we hear about in everyday life. It applies to labs, too.
Absolutely. And when disposal is unavoidable, we have to make sure we're choosing the most environmentally sound methods. Incineration is effective, but it does produce emissions. Exploring alternative treatment technologies when appropriate can really help minimize our environmental impact.
So it's a balancing act between ensuring safety and being sustainable.
Exactly. And it's a conversation that's constantly evolving as new technologies and best practices emerge.
It sounds like there's always room for improvement. What can individual lab scientists do to contribute to a more sustainable waste management system in their labs?
Well, it starts with being mindful of the waste you generate. Before you toss something, ask yourself, can this be reused or recycled? Am I using the correct container to avoid cross-contamination? Those little choices make a difference.
So it's about being aware of the impact of our actions, even something as seemingly simple as throwing something away.
Precisely. And stay informed.
Right.
Attend those training sessions, read up on the latest guidelines, and be an advocate for sustainable practices in your lab.
That's a great point. We can all be part of the solution.
Absolutely. And don't be afraid to speak up, ask questions, and suggest improvements. Sometimes those small changes can lead to big results.
I love that. This deep dive has been so eye-opening.
It has.
It's incredible how something as seemingly mundane as waste management connects to so many important issues.
It really does. It's about safety, responsibility, and our collective impact on the world around us.
Well said. I think our listeners are walking away with a much deeper understanding of the complexities and the importance of waste management in the medical lab.
I hope so. It's something we all have a role to play in.
So on that note, it's time for us to wrap up this deep dive. A huge thank you to our expert for sharing your insights and to all our listeners for joining us on this journey of discovery.
It's been my pleasure.
Until next time, stay curious.